Hey, so good afternoon, everyone. So today is our first lecture, and of course, we will be having an introduction to microbiology. So let's dissect first the word microbiology. So when we say microbiology, we are referring, of course, to the term micro, which means small, and biology, which means study of life. Therefore, microbiology is a concoction of the study of small organisms, and we call this one microorganisms. Okay, these microorganisms are too small to be seen by our naked eye. So for this particular chapter, we will focus our discussion on the introduction, the definition of microbiology, and of course, why do we need to study microbiology? And then, of course, we will be discussing, of course, the first microorganisms on Earth, the earliest known infectious disease, the pioneers in the science of microbiology, and what are the different careers in microbiology that we could have, particularly for health sciences. Okay, so as what I've told you a while ago, when we say microbiology, we biology is the study of living organisms. Okay, and microbiology is a branch of biology. So it is a branch of biology wherein it is already considered to be an advance because this one is actually more detailed and more specific for the study of microorganisms. So microbiology is the study of microbes. Okay. And these particular microbes are extremely small. When you say extremely small, they cannot be seen by our naked eye. So that's the reason why we consider all of them as microscopic structures. Okay? And some of these microorganisms could be living organisms, and some of them are considered to be as non-living entities. So our example of non-living entities are viruses and prions. So viruses and prions are considered to be as the non-living entities. As mentioned here, viroids, prions, and viruses. So these particular non-living entities are acellular microbes. So when we say acellular microbes, it means that they lack cell wall. And at the same time, they are infectious particles. So when we say infectious particles, it means that they are capable of causing infectious diseases. Okay. However, some of these microbes are considered to be as living microorganisms. Okay. So majority of them are cellular microbes, meaning to say um, they are they have cell as a basic unit of life. Okay, and of course, the most popular member of these cellular microbes are bacteria. We have the Archaea bacteria. Archaea bacteria are actually bacteria that lives in extreme environments. Okay, algae, protozoans, and some fungi. So these are examples of living microorganisms. Okay, whether you like it or not, these microbes are considered to be as ubiquitous. So the term ubiquitous means that they are virtually found everywhere. Right now, some of you are writing notes So the pen that you're holding or the paper that you are writing your notes to may contain also microorganisms, and even your skin may contain microorganisms. So microorganisms are indeed found everywhere. So that's the reason why we call it ubiquitous. Okay. So microorganisms, therefore, can be divided into the following. So we have the cellular infectious agents, and the other one are the cellular microorganisms. Okay? When we say a cellular infectious agents, it means that they do not have cell as a basic unit of life. Examples are prions and viruses. Do you know that prions are even smaller? Prions or prions are even smaller than viruses. So, in fact, they are known as infectious proteinaceous molecule, and they are the one that can cause mad cow disease, if you're familiar with this one. Okay? Or they can also cause in humans the so-called Creutzfeldt-Jakob disease. Okay, so they are even smaller than viruses. And viruses are actually divided into two. Okay, viruses may be classified as DNA virus or RNA virus. So for example, coronavirus is an example of an RNA virus. Okay, now there are also two types of cellular microorganisms. Okay, so we have the prokaryota and the other one is eukaryota. Okay, so the term karyo means nucleus. Okay, so if you hear the term karyo means nucleus. Okay, so if you say prokaryote, it means that this one has a primitive nucleus. Okay, so the term prokaryote means that it has a primitive nucleus. Okay, so the term prokaryote means primitive nucleus. Why they say it's primitive? Because these particular organisms do not have nuclear membrane. So that's the reason why we consider that as a primitive nucleus. So examples are the Archaea bacteria and the other one is the bacteria or the eubacteria. Eukaryota means true nucleus. Okay? So the term eukaryota. So the term eu actually refers to true. So ang ibig sabihin pala ng eu is true. So eukaryota is therefore means true nucleus. So examples are the algae, the fungi, and then the protozoans. Okay. So the microbes that can cause disease are sometimes referred to as germs. So colloquially, we call it germs. Okay? However, a more scientific term for a disease-causing microbes is called pathogens. Okay? So we call it pathogens. Okay, so pathogenic microorganisms. So when you say pathogenic microorganisms are the one that can cause diseases. Okay? When we say non-pathogenic microorganisms means that these do not cause disease at all. And fortunately, the vast majority of microbes are non-pathogens. So contrary to many belief, only few microbes are pathogenic. But majority of these microorganisms are non-pathogenic and they are very helpful. Okay, they have an economic importance and they are very helpful. Okay, so microorganisms that live on and in our bodies. Okay, when you say live on, it means that it is found in our skin. Okay, when you say live in, means it is inside our body. So if they naturally reside in our body without causing any harm at all, we call it indigenous microbiota. Okay, so we call it indigenous microbiota. However, some members of indigenous microbiota sometimes take advantage of hosts with weak immune system. Okay? So we call, if they take advantage of hosts with weak immune system, we call it opportunistic pathogens. Okay? So we will be calling it as opportunistic pathogens. Okay? So opportunistic pathogens will mostly infect immunocompromised host. Okay? They would infect immunocompromised host. So who are the immunocompromised hosts? Immunocompromised hosts could be people living with AIDS, or people who have cancer, because if people has cancer, if a certain patient has cancer, he's in he's in chemotherapy, and that chemotherapy can actually kill the cells of the immune system. Or people who are in life-saving transplant, people who has undergone life-saving transplant, we can also consider them as immunocompromised, because um, chances are they are also drinking immunosuppressive drugs, and these particular immunosuppressive drugs can even weaken also the immune system. So opportunistic pathogens are microbes that can cause disease, but usually do not. So they can be thought of microbes that are awaiting the opportunity to cause disease. Okay, which means that they take advantage. They take advantage of hosts with weak immune system. Okay, so this is what we meant by opportunistic pathogens. Let me give you an example. If if a person has AIDS, he'll most of the time he'll die of opportunistic infection. Majority of the patient will die of pneumonia, and then this kind of pneumonia is caused by the organism known as pneumo pneumocystis girovecii. Okay, pneumocystis girovecii. So that's the name of the pneumonia that majority of AIDS patients would die of, or would die from. And this pneumocystis girovecii is an example of of opportunistic pathogens for immunocompromised hosts. Pathogens could have two categories, namely, they can cause infectious diseases, and the other one is through microbial intoxications. Okay, so microbial intoxications means that the toxins can act as super antigen. So microbial intoxication means that the toxins could act as super antigens, and then this particular super antigen, this particular super antigen can um, can cause debilitating effects to our immune system. So examples of microbial intoxications could be caused by exotoxins and even by endotoxins. Okay? So what's the difference between infectious diseases and microbial intoxication? Okay, so let us differentiate them. So a pathogen colonizes a person's body. So the root word here is, the key word here is colonization. So colonization is somehow similar to invasion. So if the microorganisms will invade our body, so that's already considered as an infection. Okay. Microbial intoxication is also caused by infection. However, the microorganisms inside our body, okay, they will be able to produce toxin in vitro. What do you mean in vitro? In vitro means outside the body. So, meaning to say, this particular toxin can be preformed already. When you say preformed toxin, they are produced outside our body. So in infectious disease, the pathogen itself is the one that can cause the disease. However, for microbial intoxication, there is one particular step that is very important. Okay? Number one step is that you ingest the toxin, and then the toxin will cause a disease. Now, sometimes you do not ingest the toxin, but the toxin could be, you can get the toxin through traumatic puncture. So, or through wound contamination. That's another way of getting a toxin. So, the example of this one is the tetanus toxin. Okay, so you can get tetanus through wound contamination. So it's an open wound, and then your wound get contaminated by soil with bacteria, with toxin. Then that's another way of getting tetanus. So you do not ingest it, but your wound has been contaminated. Okay, so the type of this type of disease is known as infectious disease. Okay, however, tetanus is known as microbial intoxication. So examples of infectious diseases when you are infected with MRSA, which by the way stands for methicillin resistant Staphylococcus aureus, and gas gangrene, which is caused by Clostridium perfringens. Now, an example of intoxication is food poisoning, specifically the one that is caused by Staphylococcus. So we call it the staphylococcal food poisoning. Okay? Foodborne botulism is caused by Clostridium botulinum. Okay, so that one causes the foodborne botulism. Okay, so that's the basic difference between infectious diseases and microbial intoxication. So moving on, why do you think we need to study microbiology? Why is it included in nursing curriculum, for example? Okay, so of course, this one is very important, especially most of you would become registered nurses in the future, and you will be in the hospital, in healthcare facilities, wherein you will be dealing with microbes on a daily basis. So microbes play significant roles in our lives. In fact, they are essential for life on this planet. So, for example, photosynthetic algae and bacteria such as the blue cyanobacteria, and cyanobacteria, by the way, is otherwise known as the blue-green algae. They can produce much of the oxygen in our atmosphere. So that's why they are important as well. Now, microorganisms are involved in the composition of dead organisms and waste products, particularly for fungi, because fungi are considered to be as saprophytes. Okay, fungi are considered to be as saprophytes, and when we say saprophytes, these are organisms that live on decaying and or organic matter. So they live on decaying and or decaying organic matter, dead or decaying organic matter. So they use microbes to clean up toxic waste and other industrial waste products known as bioremediation. And of course, studying microbiology is very significant, especially nowadays in our most challenging times because of the pandemic. That's the reason why we're having the online setup class right now. And the pandemic is caused by COVID-19 virus, which is particularly a SARS-CoV-2. Imagine if people do not have idea in microbiology, how are we going to solve the problem of pandemic? How are we going to cope up with the pandemic? How are we going to start our research in vaccine, for example? So that's the reason why studying microbiology is very important. So here's a picture of how saprophytes, such as fungi, can live on decaying organic matter. Okay? So saprophytes and organic material are being decomposed in the soil. So imagine without fungi, we will just be seeing dead bodies lying around and they are not even decomposing at all. That's kind of scary, no? So many microbes play essential role in various elemental cycle. So we have various cycles that are necessary for our environment. To name a few, we have the carbon cycle, nitrogen cycle, oxygen cycle, sulfur cycle, and even phosphorus cycles. Okay? Algae and bacteria serve as food for tiny animals. So they can even serve as a producer in the food chain. So they are important links in the food chain. So microbes that live in the intestinal tract of animals aid in the digestion of food. So do you know that our stomach should have lactobacillus, and it will aid in the digestion of food, particularly if you will be drinking cultured milk such as Yakult. So we have the lactobacillus lactobacillus casei, the strain. Shirota strain. Okay? So if you drink Yakult, so I'm not necessarily Yakult, but any lactobacillus with any lactobacillus that are considered as probiotics, so that will also aid us in digestion. Okay? So more for many years, microorganisms have been used as cell models, particularly Escherichia coli. So the more the scientists learn about the microbial cells, the more they learn about cells in general. So that's the reason why in research, microorganisms are very important. Okay, so microorganisms are important for nitrogen fixation. So a picture here is an example of the nitrogen cycle wherein nitrogen fixing bacteria help in the utilization of ammonia. And one manifestations of nitrogen cycle is that when you pull the roots of legumes, you'll be able to see nodules. And these nodules are where we can find the nitrogen fixing bacteria. Microbes are also being used in many industries, such as in food, beverage, chemical, and even in antibiotic industries and in genetic engineering. So in genetic engineering, um, that is very important because a gene from one organism is inserted into a bacteria or yeast cell. And then the bacteria or the yeast cell will serve as the vector, and the vector is the one that receives the new gene that is capable of producing a new gene product. So, for example, we have now a genetically engineered bacteria that can produce insulin. And these are actually important for diabetic because they are not capable of producing insulin themselves. Okay? So the use of living organisms or their derivatives to make or modify products or processes is called biotechnology. And because of biotechnology, we are now able to produce food that is actually more healthy, and even vaccines are being produced as a result of biotechnology. Okay, so a picture here showing that microbes are part of the food chain because some of them are photosynthetic, hence they can serve as food for small animals. And small animals will be consumed by a bigger animal, and humans will be consuming that particular animal. Okay, so do you know the first microorganisms on Earth? So fossils of primitive microorganisms were dated back about 3.5 billion years. Wow. So the candidate for the first microorganism on Earth are Archaea Bacteria, the one that I've mentioned to you before, and Cyanobacteria. Now let's dwell on Archaea Bacteria. Archaea Bacteria are those that are, sorry, let me go back to the previous slide. Archaebacteria are those bacteria that live on extreme environment. And there are several types of Archaebacteria. This could be extremely halophilic, number two, extremely um, thermophilic, and number three are the methanogens. So extremely halophilic lives on high salt concentration. Extremely thermophilic lives on a very high temperature. So some of them could be living in volcano geysers or in hot springs or at the ventic part of the ocean wherein it is extremely hot down there. And methanogens are capable of producing methane gas. usually found in wetlands. Okay, so these are examples of Archaea bacteria. And then when we say cyanobacteria, we are referring to the blue-green algae. So infectious diseases of humans and animals have existed, okay, for as long as humans and animals have inhabited the planet. So the earliest known account of pestilence occurred in Egypt about 3180 BC. So that was a very long time ago. Okay? So the earliest known pestilence or peste. Okay? So ibig sabihin, microorganisms have lived, okay, even much older than humans on Earth. Interestingly, humans learn about microorganisms, okay, several centuries ago only. Okay, so we call it the spontaneous generation theory. Um, my slides are, there's a delay in my slides, so please bear with me. Yeah, there you go. Spontaneous generation theory. Scientists at that time were actually divided into two. Okay, those who believe in spontaneous generation theory, there are pros, and those who contradict the spontaneous generation theory, okay, we call it the cons. Okay. So spontaneous generation theory means that it is the theory of abiogenesis. When you say abiogenesis, they believe that organisms can exist, okay, from non-living organisms. So they believe that organisms can actually exist from non-living organisms. So that's what we meant by spontaneous generation theory. Actually, this is a wrong theory because they thought that we can actually exist from non-living things. Hey, so the exact opposite of spontaneous generation theory is called the biogenesis, which means that we can only exist from living organisms. Okay, so one example of that is John Needham, who experimented on chicken broth. So what John Needham did is that he had these two sets of broth. So this one is covered and this one is uncovered. So he placed chicken broth inside the tube, and then after several days, he noticed that there is already the turbidity. So the broth becomes turbid. Okay, so the broth becomes turbid, indicating that organisms have lived, organisms have existed. However, he said, how come that even the covered, this one is covered tube, has turbid organisms? Well, there is a loophole in the experiment of John Needham. And you know what's the loophole? The loophole is that he used unsterilized chicken broth. He used unsterilized chicken broth. In other words, the chicken broth that John Needham used was actually contaminated. Okay, so that's the problem with with that's the problem with the experiment of John Needham, no? So John Needham utilizes contaminated chicken broth. Okay. Now, Francesco Redi proved that maggots did not come from a piece of meat. So, what Francesco Redi did, he had this, at first, he had this two set of jar. So the first, and then he placed decaying a piece of meat. Okay. And then, and then found out that maggots appeared from the decaying meat that is uncovered, while in the covered jar, there is no maggot at all, proving that maggots did not come from a piece of meat. However, this was during the time that oxygen was discovered by Antoine Lavoisier. And of course, those who believe in spontaneous generation theory debated Francesco Redi, saying that you covered the jar and you did give exposure to oxygen. So that's the reason why there is no maggot in that decaying piece of meat. However, when Francesco Redi did another experiment this time, instead of a solid cover, he used a gauze or a mesh so that oxygen will still be able to come in, but fly will not be able to contaminate the piece of meat. And lo and behold, the results would be still no decaying meat. So that's how Francesco Redi contradicted those who believe in spontaneous generation theory. And another supporter of that was Lazzaro Spallanzani. So there, can you just imagine the unending debate so of spontaneous generation theory? So, you know, if you will liken John Needham in today's time, John Needham was like the fake news peddler because he was actually introducing theories that do not have scientific basis at all. Now, do you know how the debate ended? So, the debate ended with Louis Pasteur. So, Louis Pasteur introduced the swan neck flask experiment. So let me go back to the swan neck flask experiment. So what Louis Pasteur did, okay, he filled the flask with medium, with chicken broth, I suppose, and heated it. So he heated this particular chicken broth, okay, then drew out the neck of the flask into a long S. So he bended the flask. So it is now shaped this way. So as you can see, there is a convoluted portion. Okay? Now, because of this particular experiment, he did two sets. The first set, he did not break the neck. Okay, so this is the first set. No? He did not break the neck. And then the second set, he broke the neck. And do you know what happened to the second set? So the second set, of course, it is already exposed. So the microorganisms contaminated the chicken broth. However, in the first set wherein he did not break it, the microorganisms were actually entrapped at the convoluted portion. However, the broth remained sterile and clean. So, because of the experiment, Louis Pasteur was able to prove that microorganisms are found in the air. Okay? And because of that, Louis Pasteur became the father of microbiologists. Okay? So he is a French chemist who made numerous contributions to microbiology. He even investigated on fermentation of products. Do you know what fermentation is? So fermentation is anaerobic respiration, wherein complex carbohydrates are being broken down into a simple sugar or monosaccharide. So here, if, for example, you have a grape juice and you allow the yeast to ferment the grape juice, the product is an alcohol or a wine. However, if the grape juice is contaminated by yeast plus bacteria, the product is vinegar or acetic acid. So you do not want your wine to taste sour. So it was Louis Pasteur who invented the term pasteurization, wherein you apply enough heat to milk and juices, okay, to milk and juices to prevent contamination with bacteria. So he was the one who developed the pasteurization process, and because of the technology that was invented by Louis Pasteur, French are now known as one of the best in the world when it comes to wine making. Okay? So he discovered life form that could exist without oxygen, and by the way, we call them anaerobes. He was also the one who developed several vaccines, including rabies and anthrax vaccine. Okay, another pioneer was Robert Cox. So he was a German physician who made numerous contributions to microbiology. So he was a contemporary of Louis Pasteur. So he made significant contributions to the germ theory of disease. So this particular germ theory of disease is known as the Cox Postulate. Okay, see, he discovered that Bacillus anthracis, the one that can cause anthrax, is capable of producing spores. So he also even developed methods of fixing and staining bacteria, and he developed methods to cultivate bacteria. So it was Robert Cox who also discovered how to cultivate Mycobacterium tuberculosis, the one, the bacteria that can cause tuberculosis. So this is this is the Cox postulate. Okay, if an organism fulfills Cox postulate, has been proven to be the cause of that particular infectious disease. So, meaning to say, Cox postulate helped prove the germ theory of disease. Meaning to say, a certain disease, particularly a certain infectious disease, should have an etiologic agent. And when we say etiologic agent, we are referring to causative agent. So Robert Cox gave a tremendous boost to the development of microbiology by stressing laboratory culture and identification of microorganisms. However, there are circumstances, okay, in which Cox postulate cannot be fulfilled. So, meaning to say, there is an exemption for Cox postulate. For example, COVID-19, because SARS-CoV-2 cannot be cultured. You cannot culture the coronavirus. So that's a few exemption for Cox Postulate. Okay? So, paano ba yung Cox Postulate? How did Robert Cox discover it? So one time, while in a farm, he saw a dead cow. So how do you know that the cow is dead? Siyempre, nakabaliktad siya, tas may X. No? So he saw a dead cow, and what Robert Cox did was he actually extracted the blood of the dead cow, and he cultured the blood and allowed the organisms to grow. And then the next step is that the culture, he introduced the culture in a healthy animal and injected the culture in the healthy animal. And you know what happened to the healthy animal? The healthy animal also died from the same disease, as the cows. And when he extracted the blood of this mouse, of this healthy animal, he was able to produce the same bacteria. So same bacteria causes the same disease upon culture. Okay? So that's how the Cox postulate works. But as what I've told you, there is an exemption to Cox postulate. Number one exemptions are for those microorganisms that cannot be cultured. Second exemptions would be for those microorganisms that produces disease with similar symptoms. For example, diarrhea can be caused not just by one microorganisms, it can be caused by several other microorganisms as well. Okay, so these are exemptions to Cox postulate. Okay, so let's talk about a career in microbiology. Okay, so a microbiologist is a scientist who studies microbes. So I consider myself as a microbiologist aside from being a medical technologist. And there are many career fields in the science of microbiology. You can even concentrate on a more specific field, such as in bacteriology, the study of bacteria, phycology, or the study of algae. Protozoology is the study of protozoans. Mycology is the study of fungi. Parasitology is the study of parasites. And virology is the study of viruses. Now, for those of us who are in allied health field, our concern here is medical microbiology. So it involved the study of pathogens, the disease they cause, and the body defenses against disease. So it is concerned with epidemiology, transmission of pathogens, disease prevention measures, aseptic techniques, treatment of infectious diseases, immunology, and production of vaccines. Okay, so these are the field of medical microbiology, and of course, as a nurse, most likely you'll be involved with epidemiology because in the hospital you do not want infectious diseases to spread out as well. Okay, so that ends our lecture, and thank you very much for listening.